Today we're going to cover the process of cellular respiration. Cellular respiration is how we go through and take our sugars, and many animals take their sugars, and plants and fungus, and we take the energy that's stored in the bonds of those sugars and release it so we can use it. So the big thing to remember is we're going to start with the sugar glucose, which we've talked about. We'll take some oxygen. We're going to do some crazy chemical reactions that we're going to go over. And then at the end of this, we're going to get some carbon dioxide as waste, along with some water. But what we really want is we want that energy that was released from the bonds of this glucose. This energy we'll take and we'll store as the molecule ATP. This is the true molecule that we use to run our reactions when the activation energy. We're also going to lose some of that energy as heat. This carbon dioxide and this water, again, are waste products. You can think about it as if we breathe in to get our oxygen, because we need that for this process, and we breathe out carbon dioxide and water vapor. And that water vapor is something you'll see on a cold winter morning. We see it as we breathe out, and then we breathe out all the time. Now again, that water vapor doesn't come directly from cell respiration, but it's just a good way to remember that we breathe in oxygen because we need that for the beginning. It's a reactant. And we breathe out carbon dioxide and water because they're waste products from this cellular respiration. To break our sugars down, we're going to go through the process of cell respiration. Cell respiration is a process by slowly releasing energy in the bonds in glucose. And the glucose has a lot of bonds. If you remember the formula, it's C6H12O6. So by taking those electrons that are in those bonds and freeing them up, we're freeing up energy. And to do that, it's going to take 3 steps. So the first step is called glycolysis. Now that should sound familiar to you, or at least close to what we've talked about a lot this semester with hydrolysis. And in a hydrolysis reaction, we broke bonds. So here we're going to do the same thing. Glyco deals with sugar, lysis is to break. So we're going to break the sugar and essentially break it in half. After that, we take those 2 half parts. They're going to enter the mitochondria. Well, they'll enter the Krebs cycle. This is also known as the citric acid cycle. And again, here we're going to take and break some more bonds and free up more electrons from those bonds, because the major step that's going to give us most of our energy is step number 3, the electron transport chain. So these are the 3 major steps in cell respiration. So we're going to start off with that glucose. We'll go through glycolysis. That's going to happen in the cytoplasm. These 2 steps are going to occur in the organelle known as the mitochondria. This is known as the powerhouse of the cell, because this is where we're really going to release the energy that was in the sugar. Glycolysis is going to happen outside the mitochondria in the cytoplasm, where we begin the process. So we head into that mitochondria, and this is where this process becomes super efficient. Step number 1 that we're going to go over is, again, glycolysis. And this is going to start off in the cytoplasm. Now, if we take a look at glucose, remember C6H12O6, we're going to draw just a basic linear sequence here with those 6 carbons that are in glucose. Now, again, this is not what glucose look like, but it'll give you kind of an idea. So again, in each of these bonds, we've got electrons. It's electrons that are shared covalently that hold molecules together, and it's in these electrons that energy is stored. So it's all about breaking these bonds down and freeing up electrons and energy. So in glycolysis, this is how we're going to start off. Now, glycolysis is a very complicated process, but by the end, what's going to happen is we're going to kind of break this bond here. It's a big, complicated ninja fight, but in the end, we'll break this bond, which will free up this half of the molecule, and then this half of the molecule. And you'll see, we've gone from 1, 2, 3, 4, 5 bonds to 1, 2, 3, 4. So we've lost a bond, and in doing so, we've freed up electrons, and we'll need those. Those are really important, and we'll also free up some energy. Now, as with all chemical reactions, we need activation energy, and to start this process, we need 2 ATP. Those 2 ATP will help to get the ball rolling, and by the end of the process, the whole process will make 4 ATP. So when we take a look at glycolysis, our net ATP coming out of glycolysis is 2 ATP. Also, we're going to take these electrons, and what will happen is these electrons will enter essentially a chemical taxi cab. So that chemical taxi cab will bond with these electrons and also some hydrogens. And if you remember from the 3 steps, the last step is the electron transport chain. And that's what we want to do, is deliver all these electrons to the final one. So that taxi cab will leave and head to the electron transport chain. But if you take a look at our original kind of linear glucose, so to say, we had 5 bonds. We broke a bond, we got some net ATP, freed up some electrons. But if you take a look at this here, we still have 4 of the 5 bonds. So the energy that was stored in glucose is still primarily in these molecules here. As we want to be efficient in releasing the energy from glucose, we don't just stop with our 3 carbon molecules. Those carbon molecules head to the mitochondria, where they're going to enter the Krebs cycle, which is a whole bunch of chemical steps which are going to slowly break these bonds down and free up those electrons. And we're not going to go into depth in that, but you can think of it as this molecule entering, and we'll break this bond here. As soon as we do that, this carbon now is free. When the carbons are individual, they're going to be free as carbon dioxide. So that's going to leave the process. What's left over is a 2-carbon molecule. And again, we've also broken this bond here, so we've freed up electrons. Those electrons will also end up in a taxi, right? Just like in glycolysis. Now what will happen is we continue on with this molecule here. We'll break this bond, and now this carbon and this carbon become individual, so they too are going to leave as carbon dioxide. Here we get more electrons, and again they'll enter more taxi cabs. The same thing's going to happen with this molecule here. The bonds will be broken because it'll enter the cycle. We'll get a carbon dioxide as we release this carbon here. We'll break this bond right here. This and this carbon here, again, as carbon dioxide will leave the system. So if we take a look here in the drawing, we have 1, 2, 3, 4, 5, and 6 carbon dioxides. If you remember the formula for glucose, it was C6H12O6. There are my 6 carbons. So we freed those up, and through this process, and the major thing here is we put a lot of electrons in the taxi cabs. Right? So the Krebs cycle is going to do this, and it's going to give us a little bit of ATP in the shuffling around, but the major thing coming out of the Krebs cycle are our electrons in the taxi cab. And this is where our carbon dioxide comes from as a waste product. The third step in the process is the electron transport chain. The electron transport chain is really where we're going to harness the energy of the electrons that were in the original sugar and store that as ATP. So our taxi cabs that were generated in glycolysis and in the Krebs cycle, they're going to pull up to this electron transport chain, and this is in the mitochondria. And in the mitochondria looks kind of almost like a sausage when you see it. And then within that, there's an inner membrane. It's the inner membrane where we're going to find the electron transport chains. And we're going to have multiple electron transport chains in here. So as this taxi drives up to the beginning of the chain, this molecule here likes electrons. So it's going to steal the electrons from the taxi. Now electrons can't just be by themselves. They need to be on something, so they're going to come in hydrogens. So this molecule here will take the electron from this hydrogen and kind of discard that hydrogen that's now become an ion. So as this happens, this electron is also then going to be stolen by this molecule, which is more electronegative. And this electron gets passed down this chain, hence the chain portion of this, all the way down to the end. And as it does, it slowly releases energy. So this electron here is a high energy electron, and as it gets passed down, the energy is released, and we have a low energy electron here. But this energy is used to then pump these hydrogens to this side. As more and more taxis drop off their electrons, our hydrogens begin to build up all over this wall. And as these hydrogens build up, it's like water behind a dam. They want to go to the other side of the wall, but they can't pass through this wall except for over here, this specialized protein. So these hydrogens are going to flow through this protein, and in doing so, it actually physically spins, and the energy of that motion is used to take an ADP and a phosphate and combine them to make ATP. And this is where we get that ATP. So it's these hydrogens that flow through this molecule that generate that kinetic energy to assemble ATP. Now, as we pass this electron here, this molecule becomes happy. So if we were to continue to do this, each of our proteins would end up with an electron and be happy. And then all of a sudden this process would stop because now there's nowhere for our taxicabs to drop off our electrons. So we've kind of run into a problem. What we need to do is we need to be able to steal this electron from this final protein in the transport chain. And if you go back to the equation, and we all know we need to breathe, we need oxygen. Well, we haven't even mentioned oxygen yet. And what oxygen is going to do is, oxygen is very electronegative. Oxygen is going to steal this electron from this protein, and with the hydrogens that are flowing through, they are going to become water. So that is where the water comes from as our waste product. So oxygen is going to be the final electron acceptor at the end of this whole process. You know, if we stop breathing, we die rather quickly, and that's because this whole system shuts down. We don't make ATP, and without ATP, we can't do any chemical reactions, and we die rather quickly. In this process of electron transport chain, this whole, from one molecule of glucose, all of the electrons that will get dropped off, we will get 32 to 34 ATP. So you can see that's a large number. Now, depending on what source you use, these numbers are down, and this isn't a hypothetical perfect situation, but it is a vast number of ATP. So as a quick recap, if we take the look at our three steps, glycolysis, Krebs, and the electron transport chain, glycolysis again happens in the cytoplasm. And from this, in the cytoplasm, we're going to break the carbons, and I should say the glucose in half. We get these 2 3-carbon molecules, and we're going to get 2 total ATP, along with, again, some electrons that are free. We then move to the Krebs cycle. These molecules move to the Krebs cycle, and in the Krebs cycle, this is where we get our 6 carbon dioxide waste products, and we get a whole bunch of free electrons from all those bonds. These electrons that were in the taxicabs and on those hydrogens are going to head to the electron transport chain. Now we still do get a couple ATP from the Krebs cycle. And once all those taxicabs are dropped off here, this is where we need our oxygen. It's going to go in, pick up those electrons, and leave as water. And we're going to get 32 to 34 ATP. So we take a look at all of this. We can say that we get 36 to 38 ATP. That is for one glucose molecule. All right, so this summarizes kind of what comes out of each step in cell respiration.